The simian foamy virus is a spumavirus. Its discovery in primates has led to some speculation that the human immunodeficiency virus may have been spread to the human species in Africa, through contact with blood from apes, monkeys, and through handling or consuming bushmeat. About 70 to 90% of primates born in captivity have SFV. Animals with SFV do not display symptoms or become ill. However, recent research suggests some primates that contract SFV would become predisposed to other viruses. People who have had contact with primates can become infected with SFV. Description: although the simian foamy virus is endemic in African apes and monkeys, no evidence indicates it causes any harm to primate hosts. Its ability to cross over to humans was proven in 2004 by a joint United States and Cameroonian team which found the retrovirus in gorillas, mandrills, and ganons. Unexpectedly, they also found it in 10 of 1,100 local Cameroon residents. Of those found infected, the majority are males who had been bitten by a primate. While this only accounts for 1% of the population, this detail alarms some who fear the outbreak of another zoonotic epidemic. SFV causes cells to fuse with each other to form so-called cincture, or more figurative, giant cells, which look on a slide like foamy bubbles, hence its name. It has been tentatively linked to several diseases but without any real evidence. Co-speciation of SFV in primates. The phylogenetic tree analysis of SFV polymerase and mitochondrial cytochrome oxidase subunit 2 from African and Asian monkeys and apes provides very similar branching order and divergence times among the two trees, supporting the co-speciation. Also, the substitution rate in the SFV gene was found to be extremely slow, i.e., the SFV has evolved at a very low rate. These results suggest SFV has been co-speciated with Old World primates for about 30 million years, making them the oldest known vertebrate RNA viruses. The SFV sequence examination of species and subspecies within each clade of the phylogenetic tree of the primates indicated co-speciation of SFV and the primate hosts. As well, a strong linear relationship was found between the branch lengths for the host and SFV gene trees, which indicated synchronous genetic divergence in both datasets. By using the molecular clock, it was observed that the substitution rates for the host and SFV genes were very similar. The substitution rates for host COII gene and the SFV gene were found out to be 6 quintillion 992 quadrillion 116 trillion spade2 times 10⁻8 and 6 quintillion 992 quadrillion 170 trillion spade2 times 10⁻8, respectively. This is the slowest rate of substitution observed for RNA viruses and is closer to that of DNA viruses and endogenous retroviruses. This rate is quite different from that of exogenous RNA virus such as HIV and influenza A virus. Prevalence. Researchers in Cameroon, the Democratic Republic of Congo, France, Gabon, Germany, Japan, Rwanda, the United Kingdom, and the United States have found that simian foamy virus is widespread among wild chimpanzees throughout equatorial Africa. Humans exposed to wild primates, including chimpanzees, can acquire SFV infections. Since the long-term consequences of these cross-species infections are not known, it is important to determine to what extent wild primates are infected with simian foamy viruses. In this study, researchers tested this question for wild chimpanzees by using novel non-invasive methods, analyzing over 700 fecal samples from 25 chimpanzee communities across sub-Saharan Africa. The researchers obtained viral sequences from a large proportion of these communities, showing a range of infection rates from 44% to 100%. Major disease outbreaks have originated from cross-species transmission of infectious agents between primates and humans, making it important to learn more about how these cross-species transfers occur. The high SFV infection rates of chimpanzees provide an opportunity to monitor where humans are exposed to these viruses. Identifying the locations may help determine where the highest rates of human-chimpanzee interactions occur. This may predict what other pathogens may jump the species barrier next.